Brainstorming session about what's hot and what we think people would like to hear. That's one of the reasons the survey's out. If you know somebody want to hear a topic, let us know. And Ruth came up with the idea of the nanotechnology she had heard about.
You get two.
Nanotechnology and cancer imaging and things. And so I went on a hunt, on a Google hunt, and talked to my contacts at NIH, and I said, who's doing this, and who could come and speak to this on this topic? And they're like, wow, this is, we're just getting into this, and we're just learning about it. This one researcher here he'd be perfect for the job. And I emailed Shawn, and he was like, oh yeah, I'm available. What date? Which I'm always like, thank you, I am not worthy. So I'd like you to welcome Shawn Chen. He's NIH. His bio is like this long, so I'm not even going to attempt it.
All right, thank you, thank you. I'm like a cop with everything on the belt. Researcher. All right, so I try to be interesting as much as possible because I'm not used to facing such a wonderful audience. I'm always facing very boring scientists and researchers who have to publish papers, like saying. That's why my bio gets longer and longer over time. And this title was not made by me, it's actually made by somebody from the Rockville Cafe, Science Cafe.
That would be Tina, don't look
Tina.
at me.
So micromedicine, nanotechnology and cancer. So what is nano? Of course, it's a size. It's very small, teeny, and there's nano car, nano tartar. That's small enough, but the actual nano stuff is much smaller. Nanotechnology in general is to understand and control of materials at the nano scale. That's 10 angstrom. That's 1 nanometer. 1 micrometer is 1,000 nanometers. 1 millimeter is 10 to the 6 nanometers. So dimensions between approximately 1 to 100 nanometers, where unique phenomena enable novel applications. Because the sizes are very small, so they have very unique properties, can be utilized, can be exploited, otherwise not possible using macro molecules or macro-sized particles or materials. Right, so it's really a lot of numbers. I don't want to go through the numbers, but basically it's small. So 1 inch, 2.54 centimeters. Unfortunately, this country, unlike many other countries, still use inches and so on. So it took me quite a while. Actually, I'm jealous of this country because when I grew up in China, I never had such an opportunity to be exposed to science affairs like this. Always studying hard and day up, right? That's the Chinese English now into the Webster dictionary. So it's a lot of, that's why Chinese students, they actually study very hard, get a very good score, go to the best schools. And they recently, Washington Post is saying Harvard and MIT, they actually have higher bar for Asian students to enter the schools. Actually,
That's fair.
that's fair, exactly. All right, so how small is small? So if you look at the water molecule, H2O, it's actually about an angstrom size, less than nanometer. And then glucose, sugar, we all have in our body. And they have more in the brain. When you eat more, your sugar level in the brain will go down. And the antibody and the viral particles, actually between an antibody molecule and the viral particles, there are a lot of synthetic nanomaterials or nano devices, whatever you call it. There are a lot of things about nanopores, dendrimers, nanotubes, nanocubes, quantum dots, and nanoshells, and so on. So I'll give you a few examples later. What kind of applications these materials can do, other than you make these materials very small, but you can actually apply these materials for a variety of applications. more recently applying these materials, potentially hazardous materials, into medicine for curing cancer and other diseases. So nanomedicine is a quote-unquote the application of medical application of nanotechnology. So at least the three major applications: medical applications of nanomaterials, nanoelectronic biosensors. So you make nanomaterials, you can sense molecules at the very low level. So that's not possible by other ways. And then molecular nanotechnology. So you study nano organs or devices at the nano scale or molecular scale. So major problems with nanomedicine, of course, everything has two sides. It's hard to understand the issues related to the delivery, toxicity, and the environmental impact of nanoscale materials. So you can make a lot of things, but then there are potential issues, inhalation and then exposure to nanomaterials. What's going to happen? It's really little is done or known in the literature or in the community. So there are some hyper and hypo about the nanomaterials. And more recently, this artificial word, Theranostic. So this is actually combining therapy and diagnostics. So treatment and diagnosis coming together in one entity. Hopefully in the future, the patients will pay less, but actually will pay more because making such is so expensive at the current stage. Hopefully the diagnostics and the therapeutics come together would improve patient outcomes and safety through a more personalized approach to medicine. So this is a journal we put together, a whole issue last October, about 28 papers altogether from all the experts from the world, talking about different aspects, applications of nanomaterials in medicine and the potential problems and toxicity issues. We also came up with a new journal to discuss what is about theranostics, especially nanoparticles related to theranostic, because you have a very good surface to volume ratio. You have tiny particles, but if you have the same weight of the material, you have much more area to play with. So that's quote-unquote surface to volume ratio. You have very big surface. You can play around, put a lot of things on the surface of the material or inside the particles. And the ability of theranostics to identify novel biomarkers to advance molecular diagnostics. So in the future, for cancer, as an example, you may not put organ-specific cancer type, more of a molecular events, say breast cancer, prostate cancer, ovarian cancer. That's really, it's a very obscure term, because a cancer cell really does not recognize the organ. It's more of what molecular events leading to the normal cells to transform to a cancer cell. So it just happens to be located to an organ. That's random. But if you actually identify cancer types by the molecular pathways or the signaling or the signatures of the cancer cell, then you find a better way to treat the cancer cells. That's quote-unquote personalized cancer treatment. You know what molecular events are abnormal or actually had mutation happens to the cells, then you have a specific drug for that cancer cell, not actually the random clinical trial. At the stage you recruit patients, but unfortunately most of the clinical trials are drug-based, not the molecular signaling based. So that's actually the problem of most of the current treatments. It's not like we don't have enough cancer therapeutics. It's more about how we use the current cancer therapeutics to treat the disease. Everybody complaining about FDA is really strict, very rigid in approving any new drug, because it is true that very few new drugs are doing better than what's currently available. But anyway, so it's a little bit off the topic. Also, imaging probes and the techniques for early detection. So we have state-of-the-art imaging technologies, CT, MR, PET, SPECT, ultrasound, and so on. The technique goes better and better. However, what you see is mostly anatomical readout, not molecular readout. So really what you see, it's just the anatomical or morphological behavior of the disease, but not what's happening to the particular abnormal cells. So hopefully you have molecular probes that can diagnose and image the molecular events in real time and have a whole body readout. And the imaging guided therapy, again, you need the specific personalized imaging to guide personalized therapy, especially interventional radiology, for actually guiding the removal of the tissue or treatment or therapy of the specific locations inside the body. And then nano platforms or nanoparticle platforms that carry both imaging and the therapy components. So one shot, you will be able to diagnose the disease, at the same time treat the disease. Any questions? Of course, disease, cancer is very smart. It's before you know what's going on, and it's changing. It's constantly changing. You have a drug that's sensitive, then becomes resistant to whatever treatment because you have alternative pathways to circumvent or avoid what's going on. So you need to have a system biology approach to study disease as a system, not as an individual event or an individual cell. So it's actually evolving over time. And also, even though the United States is a very developed country, we still have people do not want to go to the hospital. Most people don't want anyway. So you need the point of care technology or strategies to allow patients to stay home, have a better quality of life over time. So then you really have to personalize the devices and care to be done at home, not at the hospital, and also at much lower cost. Talking about healthcare reform, you really need to have to lower the total cost. But that's actually debatable because drug companies don't want that happen. And, and also drug company may not want the true personalized treatment to happen because if you have only a small percentage of the patients are likely to respond to a given treatment or given therapy, a given drug, then the overall market would be smaller, even though the effectiveness would be higher. So that's really the dilemma. But then I think both industry and academia are thinking, try to find a striking balance between the margin market and the actual social health benefit. Well, let's put all this blending together. Actually, there is a company called Blending. It's Bob Langer from MIT. He's one of the 10 scientific advisors for Obama administration. So he has a new company called Blending. Just mixing everything together, then it will do a lot of things, cannot be done separately. So you have nanotechnology, put the imaging and the therapy all together through nanoparticles. And the recent chemical engineering news also highlight the importance of personalizing nanomedicine and put a few examples. This, of course, most of these cartoons are hypothetical and very cartoon-like, of course. So it's not there yet because it's rather complicated. You have multiple components in one system, then going through the hurdle of FDA approval for clinical trial. So you have to test the toxicity and the safety of each component and the combination of different components coming together, which is non-trivial and very costly. So I'll give you a few examples of nanomaterials have been used for at least at the preclinical level, meaning either at the cellular level or small animals, rodents, large animals, pigs, dogs, primates, to test different nanoparticles. So for example, the light-emitting quantum dots, gold nanoshells, gold nanoparticles, very old-fashioned liposomal nanomaterials, iron oxide, then iron cobalt, ultrasound microbubbles, carbon nanotubes, dendrimers, and so on. Some of them probably very familiar to you, some of them are not necessarily very familiar. Most of these materials are inorganic metals or semiconductor materials, so which was not originally used for health-related studies, because these materials are considered toxic. Because imagine you have, inert metal or semiconductor materials inject to the body, what's going to happen? But there are ways to avoid that. That's modification of the materials, make it biocompatible. So, the human body or animal body would not recognize the material by itself, but would most recognize the surface coating. So that way would fool the cells around that you don't really the cells won't know what's going on with the particles, but the particle does have its own intrinsic physical and chemical properties allow imaging. For example, quantum dots has light emitting properties. You can use optical imaging to look at the particles distribution and the fate of the materials inside the body. So I'll give you one example of quantum dots and one example of iron oxide and one example of carbon nanotube later. So 3 examples. If you want to use materials for drug or gene delivery, you have somewhat different structures, because you need to have the particles like a sponge to load the material or load the gene construct, whether it's DNA plasmid or srRNA, microRNA, oligonucleotide. Traditionally, gene therapy is based on viral particles, virus. So even though it's not the real particle, but the people call it a virus particle, it's also the size of the nanoparticle. But the virus has its own problems, all the immuno problems and other problems. So really most of the lentivirus, adenovirus-based therapeutics in the clinic are really on hold. But more recently, how to use nanomaterials to replace those virus and other drug carriers. For example, paclitaxel, that's an anti-cancer drug. The currently mostly to make these very lipophilic or hydrophobic molecules soluble or be able to deliver under physiological condition, you have to use a chromophore. It's very painful to solubilize the material they inject. It's very sticky. And then more recently, they use albumin simply to mix that albumin protein with paclitaxel, so-called Abraxane. That makes really water soluble, and it's much better, even though actually it's now in real shortage. If you Google Abraxane, then you'll see Abraxane shortage. Yeah, so a lot of polymer-based materials or self-assembled particles allow carrying drugs at different locations, inside, within, or on the surface of the materials. So then the loading efficiency is definitely very important. So quantum dots, because the dots have very brilliant colors, and the color is dependent on the size. When the size of the particle grows bigger, it actually redshift. So you have the UV light or blue light all the way to the near infrared or even infrared light. So that becomes possible, and you can fine-tune the size, and you will have many different colors, and you can also mix these particles of different sizes, make a really many labels. So far, you are able to create up to 40,000 labels. So very much like a painting process. You can actually put different colored pens, mix together, like an artist doing that. And this can be done with quantum dots. So that's what's been seen when you have UV light shining the material in the dark. You will see this fluorescent light coming out. So you have some representative colors here, all the way from short wavelengths to long wavelengths, and actually going here, then it won't be visible because naked eyes won't see near infrared and infrared light. But so what we have done, or many others have done, is to make these particles soluble in water. What you do on the surface, you coat a amphiphilic polymer because these particles are lipophilic with surfactants on the surface. So then you have an amphiphilic molecule, which means one end is water soluble, the other end is lipid soluble. So you can intercalate these amphiphilic molecules, one end interacting with the surfactants on the surface of the quantum dots, the other end is sticking out, it's water soluble. So that way, these particles becomes dispersible, or actually not precipitated, but almost like a solution. It's not actually a solution. It's a well dispersed in aqueous solution. Then you do some kind of chemistry, put the targeting molecules. In this case, it's a peptide that would recognize a receptor on cancer cells. For example, integrin alpha V beta 3, that's a specific receptor. And these particles are not perfectly spherical, they're actually ellipsoidal, about 25 to 30 nanometers in size. And this is the atomic force microscope, AFM, measuring the individual particle shape and size. So that's what the particle look like. And there is the halo around the particle. That's actually the polymer coating. So you do have a little bit on the surface, allowing it water soluble. What's inside won't be recognized by cells, but it does allow the particles to be freely circulating around the body and then distribute to the locations it should go. But like many nanomaterials, because of the size, it's bigger than actual protein or small molecules. They would be engulfed by macrophages in the body. The RES organs, reticular endothelial system RES organs, especially liver, spleen, bone marrow, joints, lymph nodes, and so on, they would be able to capture all those nanomaterials or large particles, micro-sized or nano-sized particles. That's why you see here from imaging, you see, even though this a little lousy optical imaging, the green color is a pseudo green. It's actually coming from the autofluorescence. This is a little mouse. A mouse skin is very fluorescent. If you actually put either a biopsy mouse or a nude mouse in the dark, you won't miss it. It's actually glowing a little bit because the skin and the fur are actually fluorescent. So you will see those. So that autofluorescence can confuse with actual fluorescence coming out of these nanoparticles. But with the mathematical algorithm, you can separate this color because they have different different optical spectrum. You can separate the spectrum. And this pseudo red color is coming from the quantum dots. You see really a lot in the liver and bone marrow and joints and so on. So that's indeed true for this type of materials, but also it does accumulate specifically to the tumor area. Of course, this tumor model is very artificial. You get a immunodeficient mouse, then you have immortalized cancer cells grown in cell culture. Then you inoculate those cells under the skin. These tumors would grow very healthy into a spherical size. And these tumor cells, somewhat mimics what's going on in the clinic, even though it's very artificial. But it does express the certain receptors, in this case, integrin alpha V beta 3, and this RGD peptide conjugated cadmium telluride quantum dots would go here, but the many more also go to the liver and spleen, which is unwanted. Yeah?
So you are targeting it by using RGD
Peptide, yeah.
peptide, but RGD is so common, so how are you targeting
Right.
specifically tumor?
Yeah, it's a very good, very scientific question, right?
Could you repeat it?
Yes, so the question is, the RGD peptide supposedly is to target the specific integrin, but her question is, there are so many different RGD peptides. How come this RGD is specific for this particular target? Indeed, RGD, so it's arginine, glycine, aspartic acid, 3 amino acids. So it's an abbreviation of many peptides containing these 3 amino acid residues. They're called RGD, but the proteins or peptides with RGD sequences can have very different configurations. Different configurations, different structures would allow different binding specificity and affinity for different integrins. So this particular case is actually this one is a head-to-tail cyclized RGD peptide. So that way it's specific for alpha V beta 3, and it does mimic the binding of vitronectin protein. Sorry, it's probably going a little too much.
You two knew what you were talking about.
Right, so let's move on to carbon material. So a lot of people have, views and enthusiasm about carbon nanotubes. You have single-wall carbon nanotubes, multi-wall carbon nanotubes, nanodiamonds, C-dots. There's so many different terms. And you go to Wikipedia, you will find a lot. So for single-wall nanotube, of course, a lot of physics about it. I don't have to talk too much because the focus of this talk is more on the application of these materials, similarly irrelevant materials for medical applications. So it has a very unique optical properties, especially Raman properties. It has several very distinct features, so-called radial breathing mode, RBM mode, then D mode, then the high energy G mode, G prime mode. So this peak from Raman spectrum can be used for imaging. First of all, like quantum dots and many other nanoparticles, the first is to fool the cells around, make the water soluble, less recognized by macrophages. So in this case, again, it's a carbon suit you can buy from many companies in a very cheap price. What you do, there are many ways, of course, to make the water soluble. One of the methods is to use a phospholipid to coat the nanotubes. Because the tubes has a lot of unique properties. First of all, it's very lipophilic. Second, it has a very conjugated surface or pi surface, sp2 carbon. If you have studied high school chemistry, physics of carbon, there is sp3 and sp2 orbital, and in this nanotube is sp2, so it's all pi conjugate system. Then any molecules that actually has pi system, for example benzene, other molecules would be able to actually have the pi-pi stacking on the surface of the nanotubes. In this case, we put the phospholipid, that's a hydrophobic interactions between this alkyl chain and the carbon surface. Then you put a polyethylene glycol that make it water soluble. Then at the end of this polyethylene glycol, then put different things. So what we again use the RGD peptide as a model peptide to couple onto this nanotube, and then we also label this nanotube with the radioisotope, copper 64 in this case. Actually, non-radioactive copper is copper 63. Then you have many radioactive copper isotopes. Copper 64 is a positron or positive electron emitting radionuclide, can be made by a cyclotron, medical cyclotron. So you label this nanotube with isotope, Then you can do a PET imaging, positron emission tomography. Actually, if you go to a hospital nuclear medicine department, there is actually a big PET department. So now becomes routine to scan patients for potential cancer and other neurodegenerative diseases. Because this G-band is very unique, so you can, and that's not, it's only for nanotubes. Any tissue in the body won't have such Raman signature. So we can use this to quantify the distribution of the material inside the body. So of course for mouse, we can sacrifice the animals after injecting the material to the body, then use the Raman spectrometer to measure the Raman intensity, then quantify, convert the numbers into percent injected dose per gram or standard uptake values, whatever. Also, because of the Raman signature, you can use a Raman microscope to look at the distribution of the material directly, even though at this point we still don't have a whole body Raman scanner because the detector is a single detector, not an array detector. So it's very low speed detecting. So if you have a microscope, it's very much like a fluorescence microscope or confocal microscope, but they actually can be combined. You can look at the Raman. You do a raster scan, really the pixel by pixel scan. So, for example, this little area of the tumor area, 5 by 5 millimeter, will take us about 20 minutes for the moment, be able to scan this, what's happening here. Of course, you see these shining spots, very relatively low resolution because it's really just pixel by pixel scan. But you do see difference. When you have a specific targeting, the nanotubes do go to the tumor area. Without a specific targeting, these nanotubes in the tumor areas have very little to no background signal. So you can semi-quantify the signals in the tumor areas. It's very obvious, almost black and white difference. So even though these materials are not necessarily to be translated into clinic for patient care right away, but they do hold high potentials because these materials do have very unique properties that are not possible based on the current techniques. Or most of the contrast agents in the clinic are small molecules. For example, CT contrast agent is iodine based. It's really the I don't know anybody has done contrast enhanced CT. It's really syrup-like, very sticky. Liquid very painful, and yet the contrast very poor. And the MR, you have gadolinium DTPA. It's also small molecules that's also toxic in a way. So antibodies, this also have another property because it absorbs light and emits heat. So that's a photoacoustic property. So it really, you have nanotube absorbs light, then the tissue having, or the environment with this material after absorbing light, there is definitely thermal expansion. Especially tissue would have much better thermal expansion than the many other materials. This expansion would produce acoustic wave, and this acoustic wave, of course, can be detected by a ultrasound transducer. So that way, unlike ultrasound, it's a sound goes in, sound comes out. So here is light comes in, and then sound comes out. So that's the so-called photoacoustic imaging. And there are already commercial devices available to detect photoacoustic signal. Unlike optical imaging, even though the detection is light-based, but unlike light, which has limited penetration into a tissue. For example, when you have a laser light shining to the finger, you have the red light, you probably see the red on the other side of the finger, but that's pretty much that, about a centimeter to 2 centimeters you can go through. But then when the emission light comes from the other end to the same surface, there's another attenuation, a scattering of the light. So detection efficiency is very low. Coming to photoacoustic, so at least solve the problem halfway. You have the sound goes in, sorry, light goes in, that's the same penetration, but the sound comes out that has a little attenuation compared to light, so would have more sensitive detection. And these nanotubes has unique properties, so we can actually combine ultrasound and the photoacoustic imaging. So this one, it's an ultrasound. Really, two tumors before injection of the nanomaterials, they don't see any difference at all. And then before injection of the contrast agent, you see a little difference, but you do see some background. And this is actually the probably when you have the hemorrhage, they have hemoglobin and deoxyhemoglobin with different ratios. Then what actually here, both hemoglobin and deoxyhemoglobin have photoacoustic properties because they are colorful, they do absorb light. And then post injection of this particular contrast agent, you see difference, and then you subtract this background, you'll see an absolute difference. With the targeting, you have a very good signal coming from the tumor area, and without the targeting, you don't see the tubes accumulate in the tumor area. So, and then you can cross-validate these findings by Raman imaging. Yes?
How big is this tumor that you need this photoacoustic?
Because the resolution of photoacoustic is pretty high.
No, so that's I'm asking, how small is this tumor that you cannot detect it otherwise except photoacoustic?
That's a very good question. Even though this study is very artificial, you have the tumor. The question is how small the tumor or how big the tumor has to be so that the photoacoustic imaging can detect the tumor. Because why do we need the photoacoustic imaging, right? Yeah, so you have many ways to detect tumors at a very small size. For example, MR and the ultrasound and the CT, they do detect tumors at a small size. So what we are talking here is really not how small the tumors can detect, because that's still talking about the anatomical size. But what the anatomical imaging doesn't tell you is what's going on with a small tumor or a big tumor. So what's happening? So then what will be the treatment to go for a small tumor or big, mid, or large-sized tumor? So hopefully this kind of probes will tell you there is a certain abnormality going on in the tumor that either whatever imaging method you want to choose, that's less critical. It's more of how you can sensitively, accurately picture the molecular events or the receptor or enzyme distribution on the tumor area.
So this is actually the theranostic application?
Yes, potentially, yes. So you have not necessarily, but it can be, right? You can have this material actually carry drugs. We actually have several studies. It does load a lot of different drugs to allow a very interesting therapy because it does accumulate in the tumor area. So now when you have this material also carrying drugs, you want to have a better delivery of the drug than the free drug alone in actually homing to the tumor area. Then of course, the hypothesis when you have more drugs accumulate homing to the tumor area, then you would have a better treatment efficacy. Because most of the drugs, unfortunately, after IV injection, it has less than 0.1 to 0.5% of the total drug actually acting on the cancer cells. Most of them get stuck in the major organs and produce toxicity. Really, most of the cancer therapy, especially chemotherapy, they're just too toxic. The patients really suffer from the toxicity, not from the benefit of the efficacy of the treatment. That's the main problem. So all these different carriers or delivery vehicles would allow better and more efficient or sometimes smart delivery of the drugs. Yeah, if you have the drug that's actually not toxic until it hits the target, that would be better. So even you have drugs, prodrugs, so-called, located elsewhere, but it won't be activated or becomes toxic at all. So then it's much better than a free drug being injected. And then of course, we have other ways to further improve the sensitivity, so I just probably skip this. And end with this magnetic nanoparticles. The reason we want to spend more time on magnetic nanoparticles, this material is iron dust-based, iron oxide nanoparticles. So you can make these particles very small. You can, for MR imaging, for stem cell labeling, for multimodality imaging, for drug and gene delivery, and for activatable probes. So I give you examples how this material is being used for imaging cancers. To answer this latest question again, MR has almost the best resolution so far. And if we also allow MR imaging to be able to dissect the molecular events, then it will be even better. So what we are doing is to have iron oxide nanoparticles coated with polymer, make it water soluble, like many other nanomaterials. First to label, to conjugate these particles with different materials, make it biocompatible, quote unquote. And these particles with very nice uniform size. Then we did the MR imaging in a small mouse, so the resolution is definitely very good. You not only see the, so this iron oxide nanoparticles without targeting, and this iron oxide with RGD peptide technique. And so tonight I only use this RGD peptide as an example. It can be coupled with many different things for different purposes. So in the pre-injection, you see this contrast, the artificial tumor. Then the post-injection, without targeting, these particles don't go there. So there is not much signal change at all. So here, this color, of course, it's a pseudo color. So the, from here, you have a high signal, and here is a low signal. The iron oxide gives a negative contrast. So that's why you see signal reduction over time. And also you notice in the tumor area, it's not homogeneously distributed. It's heterogeneously distributed. Some, the tumor, unfortunately, when it grows into a certain size, it becomes very heterogeneous, and some areas are less permeable or necrotic. So then these drugs or any materials won't go there. Same thing happens to nanomaterials. So areas with signal reduction are the particles being distributed. Areas, this, for example, here, even some of the surface areas and some in the center won't have particles accumulate. Then these areas, unfortunately, becomes drug resistant. Even you treat the cancer, most of the cells being cured or killed, then you still have some residual tumors remaining, will never be actually hit. Then these tumors may become very active, regrow, and relapse, then metastasis, and so on. So that's actually the real danger of cancer treatment. A drug is effective if initially, but later on becomes resistant. It's both either the cancer cells becomes resistant, or some cells was never hit by the drugs or the molecules. So the same material, like we call it Theranostics, because not only it allows imaging, but also allows carrying drugs at the same time. So the same material, like here, you can image, but at the same time, you can carry drugs. For example, I use doxorubicin as an example, which is a fluorescent molecule, but also it's an anti-cancer drug. We can load a pretty good level of the drug, and then it's pretty effective, so I don't want to go through the details. But basically, it's interesting that this free drug versus nanoparticle carrying the drug would have very different behavior internalizing into the cells. For example, free doxorubicin takes time. So this blue color is a cell nucleus to identify where the cells are, DAPI standing. Then the red color is coming from the drug, doxorubicin. So it takes time for the drug goes into the cells and then locate to the cell nuclei. For the nanoparticles, it has immediate internalization. So they have two different mechanisms entering the cells. One is through the lipophilic penetration through the cell membrane. The other one is endocytosis. So you have the nanoparticles gobbled by the cell membrane right away, get into the cells quickly. Then the drug get released from the particles. Then it actually finds its way to the nucleus. Also, we tried the different cancer cells, the ovarian cancer cell line of CAR8, and then this drug-resistant cells. So if the free doxorubicin for the drug-sensitive cells, the drug does go in the cell to the cell nucleus, then kill the cells. But then if it's drug-resistant, so how you make the cells drug-resistant? You treat the cells with low dose of the drug over and over, then the cells will actually adapt to the environment, becomes resistant to the cells. Actually, this drug will not go in the cells at all. Will not go in. That's why it becomes drug resistant, because the drug will never hit the cells. And it was always in the solution, never enter the cell. But coming to nanoparticles, drug resistant cells, so the drug still goes in pretty effectively. So that's one of the major advantages of the nanomaterials. Not only you allow better delivery, but also better homing to the locations of the cells or organs of interest. Then we can do imaging, you can do therapy. Actually, really it's a little bit complicated. It's a growth curve of the tumor model. You see, using this formula after several injections over time, you see a much delayed tumor growth compared to the control group, that without treatment the tumors grow very big and the mice have to be sacrificed. But then when you have a nano formula, it's much better. I'm not sure whether it's too much for the audience. Yes?
The injection, is it an intratumoral injection, or is it more of a...
No, intravenous injection, not the intratumoral injection. If it's intratumoral injection, both drugs would behave very nicely. But the intravenous injection, then you have to have the drug home specifically to the tumor area, because the drugs elsewhere won't be useful at all. Yes?
Does it matter how big the drug molecule is for the success of the therapy? I mean, are there drugs that are too big to get even with this technology?
Yes, the question is what is the size effect of the drug or particle formula to be effective, to be able to locate specifically to the tumor area? Actually, there are some theories about the ability of a material to locate to the tumor. One is so-called EPR effect. It's enhanced permeability and retention. Because tumor area, unlike the normal tissue, they have very leaky vasculature. So you have the particles of a certain size, they actually get into the area, but then they won't leak out easily, like small molecules. So that way, you have, even you don't have specific targeting, this material can still have a better homing than the small molecules. The other way is specific targeting, but still the size is a real big factor and have a major impact in the distribution, the redistribution of fate of the material inside the body. If the particles are too big, after intravenous injection, if it's micro-sized particles, then you get stuck in the lung. So then really, once it's in the lung, it's very difficult for these particles to come out. Actually, think about stem cell therapy. Cells are actually micron-sized particles. If you think that way, you do cell therapy, T cell therapy, or mesenchymal stem cell, or embryonic stem cell therapy. If it's an intravenous injection, all the cells get stuck in the lung immediately. But it takes many hours for the cells find their own ways to slowly coming out, then locate to the areas of possible locations. But for nanoparticles, unlike cells, they are not as smart like cells, so they are not live. So then once they're stuck in the liver or in the lung, then they will be difficult to sequester out of these organs, recycle to the circulation. So it's very difficult. So first of all, the particle cannot be too big. If it's too big, then it's defeating the purpose. cannot be too small. If it's too small, then the loading efficiency will be very low. You want to have many drug molecules to be loaded onto a single particle, then it really does not serve the purpose. So generally speaking, it's for imaging purpose. Imaging, only you want to see the picture, then you want the particles get out of the body as soon as possible. So you want smaller particles. So you allow the particles to hit the target, then clear out of the body, especially renal clearance through the kidneys to the urinary bladder, or sometimes through liver to the gut to feces. For particle carrying drugs, then you need the size somewhat bigger to allow longer circulation half-life, to allow the particles to balance itself inside the body, finding its way to the target, then slowly releasing the drug, not the burst of releasing the drugs. Because when you have the drug hit the target right away, then you have a very immediate effect, but the cells need to sustain treatment before it can be killed. So one of the particles with a given size, typically 50 to 80 nanometers. It's not, of course, sometimes smaller size will do a better job, but typically that size, less than 100 nanometers in general, to allow good circulation. And also it's the art of chemistry, how to make these particles metastable. Not very stable, so then drug will never release, but not very broken right away, so then drug get released too quickly. So it's really quite a challenge. So to quickly summarize, so ideal nanoparticle for imaging and therapy would allow very selective and rapid accumulation in the diseased site, report biochemical morphological characters of tissues and organs of interest, deliver effective therapy, and safe, biodegradable, with non-toxic byproducts and lack of immunogenicity. It's a really long sentence, and it's a very high bar. It's very difficult to make the dream come true. Because of the complexity of the theranostic nanoparticles, because you have the delivery vehicles, you have the cargo, you have the targeting ligands, you have the imaging labels, you have the drugs, or drugs of the quote-unquote the cargo. Specifically deliver everything to specific tissues, cells, or subcellular components. Translation of this into clinic is a very daunting task, for sure. It's non-trivial, it's very costly. That's why we have invested big money for very small science. So here we are talking about the all right. Well, even though a lot needs to be done, but the enthusiasm remains, because it's challenging. It's hopefully in the near future we'll find smarter ways to make it simpler and yet effective. Yeah, go ahead.
I've got a question. My background is physics, and I always struggled with organic chemistry.
We struggle with physics.
So I'm trying to understand this. From what I'm understanding, these nanoparticles are inorganic. Is that correct? And
most of the nanoparticles for imaging applications are inorganic because they have the physical properties allow them for imaging purpose. But for drug delivery, not necessarily. Many of the nanoparticles are self-assembled based on the polymers or the proteins. There are many proteins actually self-assemble into particles. For example, ferritin, that's actually a very beautiful spherical particle with 24 units of protein molecules coming together with actually a hole in the middle, and it's a nano shell by itself.
Well, how do you get these nanoparticles to go to the targeted area?
Right, so several things we do. First of all, to make these materials, they are quote-unquote inert, and then make it water soluble. It has to be soluble in water, in blood, under physical conditions. So then of course, what you do, you coat these materials with water-soluble molecules or amphiphilic molecules. That one part that actually sticks with the particle, one part makes it water-soluble. So it's really fooling the surroundings to for the environment to recognize the surface only, not what's inside the particle. Then you conjugate or combine some targeting molecules, so whether it's antibody, protein, peptides, or sometimes small molecules like folic acid, for example. You can couple these molecules onto the surface. So it's a simple chemistry there. Sorry. So this really the ligand would recognize the receptor or enzyme. These particles are really being tagged by these ligands to go to the target. So it's really like a magic bullet approach, which yeah.
That's how I always thought they did organic chemistry with a magic bullet.
I first thought you were designing the nanoparticles to find the cancer cell, but really what you're doing is you're using the nanoparticles to carry what's finding the cancer cell.
Yes, in a way it is. So that's precisely majority of the nanoscience in medicine. That's what it does. Really, you have a nanoparticle. Really, nanoparticle is just a buzzword.
Well, but the medicine gets lost. But once you attach it to the nanoparticle, it keeps finding its way.
Yes.
Actively.
Yes.
Okay.
Yeah. That's at least the hope. At least the hope.
So what's the advantage over monoclonal antibody delivery?
Versus nanoparticle-based delivery, right? For example, Genentech is spending big money into the antibody conjugates, right? So they actually, for example, the Avastin,
right?
You know, and now it's really off the market for breast cancer treatment. But what they are doing, they're actually now using such antibodies, including Avastin, Herceptin, and other antibodies, to conjugate drug molecules or therapeutic proteins onto the antibodies. So there are several issues with those antibody, of course, so-called biologics. Now the bioengineering technique is really also advancing very fast, so they can make these antibodies into very large scale, and the cost for the patient to go to a drugstore to buy this drug, the actually the manufacturing cost is the actual drug cost for the patient is 5,000 times more than its production cost. It's amazing, but the cost of the, of course, all the way from concept to the market takes a lot of money. So then it's really, you cannot, compare that. The same thing with nano. We hope in the future we can reduce the cost. At this point, it's still quite expensive to make nanomaterials into large quantity, into kilogram or tons of nanoparticles. Think about the size. Then, of course, the size of the typical nanoparticle would be similar or somewhat larger than antibody. But antibody, if you over-modify the antibody with drugs, then the antibody would lose its ability to recognize the antigen on the surface. But nanoparticles, on the other hand, it's just a carrier. So you put a lot of things on the surface, it does not necessarily affect, because you can still put antibody on the surface of nanoparticle, and then it's just a mono-modification, just the one side of the antibody to be modified. But if you have to load a lot of things on the antibody, many sites on the antibody would be modified, then it loses the effect. Yeah. So I wouldn't say which is better, because they serve quite different purposes. Yeah. Yeah.
If I understood correctly, the photoacoustic has a heating step after the photon gets absorbed before it gives out the sound.
Yes.
The heating effect itself be used against tumor cells?
Yes, that's a very good question. The question is, for photoacoustic imaging, not only it's being used for imaging, but also the photothermal effect can also be used for therapy, right? So nanoparticles, for example, gold nanorods. So it's really just a rod-like gold particles. When you shine laser onto the particle itself goes into several hundred degrees instantaneously. But of course, you don't get the tissue surrounding it to get that high temperature because you have a heat dissipation over time. There's physics around it. But the tissue do go to a reasonable high temperature, but not too high to, if it's for imaging purpose, you don't want to damage the tissue. So you don't want to have the tissue temperature go higher than 42 degrees. But if you have a larger dose of the same material for therapy purposes, then you could heat up the tissue up to 60 or even higher degrees. This, of course, it's very different from the thermotherapy. Thermotherapy or HIFU use high frequency ultrasound treatment for prostate cancer, for example. That way, it really is cooking the tissue, but it's a physical cooking of the tissue. So it's not homogeneous. You really have the tip poking the tissue, then you really burning the tissue. And then you have always immediate color change of the tissue. Actually, if you have seen in the animal studies or in the clinic, but then the surface of the tissues are not necessarily being treated well, and you only actually cook about 70 to 80% of the tumor area, and also normal tissues got affected. But if you have nanoparticles homogeneously distributed, then you don't have such a problem of uneven heating, because particles supposedly being homogeneously distributed throughout the tissue, then the whole tumor area, not the normal tissue, get heat up. So that's the potential advantage of this photothermal therapy over the traditional HIFU treatment.
Question. The quantum dot, is it continuously giving off light, or does it need to be shot with a laser or something like that?
It does if you actually, well, you need the laser all the time. But the advantage of quantum dots over fluorescent dyes, organic dyes, it does not photo bleach. So fluorescent dyes, if you keep shining laser over a minute, then it's really photo bleached. It won't emit light at all because the molecules got destroyed. But the semiconductor quantum dots won't be. So it actually can stay for many hours. Keep shining laser, can still emitting light.
So prior to injection,